There is a surge of interest in degradation-based therapeutics, such as protacs and molecular glues. How does your work on CRL biology inform the rational design of the next generation degraders? And what do you think is the most misunderstood challenge in bringing targeted protein degradation to the clinic?
In the 90s, people were very excited about discovering that they could target protein kinases. And although in most cases you are targeting the catalytic site of the protein kinase, the ATP binding site, different companies were able to identify specific enough kinase inhibitors that reach the clinic. So once we understood that the family of ubiquitin ligases was larger than the family of kinases, most of us thought, why don't we target ubiquitin ligases like we target kinases? And the truth is that, to make a long story short, it was much more difficult. And none of what different companies or even academic labs did to target ubiquitin ligases went too far. And so people basically lost interest in the ubiquitin system from a therapeutic point of view. This until people created PROTACs. So PROTAC stands for proteolytic targeting chimera. And the original discovery was made with a fusion protein. It was made by Ray Deshay and Craig Cruz. And so what they did was to fuse two domains. One domain of a protein that binds and one domain that binds a protein substrate with a linker in between. So now you have this small protein that at the same time can bind a substrate and an ubiquitin ligase, and artificially brings together a substrate that normally doesn't see that ubiquitin ligase and vice versa together. So once they are together, the ubiquitin ligase will start to ubiquitinate the new or neo substrate, the neomorphic substrate. That substrate will be degraded. So this was the base of molecular products, where now you have small molecules. You have two small molecules with a linker in between. Rather than two protein domains, you have two warheads. One binds the E3 ubiquitin ligase, the other one binds the substrate. And these products are able to bind, to bring together substrates that normally are not ubiquitinated by certain ubiquitin ligases and make them substrates of these E3s. So small molecules, or relatively small molecules like proteins, are used now in clinical trials to eliminate bronchogenic proteins or proteins that are involved in diseases through some ubiquitin ligases. Now the importance of that is that thanks to proteins, you can eliminate proteins that don't have a catalytic site, so proteins that are not enzymes. Because in most cases, it's easier to create an inhibitor of an enzyme, a small molecule that interferes with enzymatic activity of a protein.